Hi, this is Professor Paul Knopfler here at UC Davis School of Medicine. I'm a stem cell researcher, but I also have been running a blog and website of resources called The Niche for more than 10 years. And I'm adding a new series of videos that kind of provide educational resources to people, also in some cases fact check things. Today's video is actually more about the sort of basic science of stem cells, and the topic is embryonic stem cells, and especially human embryonic stem cells. So I'm going to go ahead and share my screen with you. Let's see. And kind of go through this post I've written about some of the key things you need to know about embryonic stem cells, and for instance, where do they come from? So this is my site, The Niche, and you can just kind of scroll down in this article, and you can see the different things, the different topics in this particular article about embryonic stem cells. So perhaps what we can do is start by asking where embryonic stem cells come from. And with only a few exceptions that I'm going to talk about in a minute, embryonic stem cells, as their name suggests, actually come from embryos. So in the case of human embryonic stem cells, what this means is that you need some embryo as the source to make your embryonic stem cells in most cases. So these embryos are sort of special embryos in a sense, in that they're usually left over from fertility procedures or IVF, in vitro fertilization kind of procedures that are just left in liquid nitrogen. Because lots of times when fertility treatments are done, the fertility doctor makes too many embryos and those are just left. So unfortunately, a lot of those embryos, because it's expensive to store them in liquid nitrogen, are just discarded at some point. So instead, in some cases, they'll be used to make embryonic stem cell lines, which can have a number of different uses that we'll go through today as well. So once you excuse me, start your embryonic stem cells from an embryo, most of the time, they'll actually sort of, as you're growing them in culture, interestingly, they almost sort of try to develop that same environment like they have in the embryo. So you can see in this picture, this is not one embryonic stem cell. This is actually a colony of embryonic stem cells all kind of clustered together. Probably a few hundred cells here kind of making this circular structure. And they like to all be kind of packed in tight together like they were in the actual embryo. Embryonic stem cells are really sensitive cells. So sometimes we'll actually grow them on top of other cells. So you can kind of see underneath this colony, if you kind of imagine it having a little bit of a three-dimensional structure, there are these fibroblast feeder cells that secrete growth factors and extracellular matrix factors, basically just making a nice home, like almost like a couch for these embryonic stem cells all clustered together in this colony. So the embryos that are used to make embryonic stem cells typically are called what are called blastocyst stage embryos. So in humans, they have only about 100 cells. And they look just kind of like a little round circle of cells, and I'll show you that in a bit. Embryonic stem cells belong to a class of stem cells called pluripotent stem cells, and this just means that they can make many different kinds of differentiated cells. So there are a couple of ways you can make embryonic stem cells without actually using these IVF embryos. So more rarely, sort of part one in this blog post is something called SCNT, or somatic cell nuclear transfer. This is sometimes called cloning of stem cells or therapeutic cloning. So what happens in this case, and the reason we talk about the cloning part, is that in order to make embryonic stem cells, you actually, instead of using sort of a regular embryo, you actually take an egg and you remove its nucleus and you put in the nucleus of what we call a somatic cell. So this is just an everyday cell in your body, like a skin cell or something like that. And then in some cases, that egg now, or sometimes it's a one-cell embryo that now has its nucleus swapped out and a new nucleus swapped in, will actually start growing as though it's an embryo. And you can then use that to try to make embryonic stem cells. And this has actually been shown in a number of different research papers. It's kind of difficult to do, much more difficult than just using a typical embryo. The more rarely part two, the other kind of way you can make embryonic stem cells without using up an embryo that comes from IVF is using an earlier stage embryo. So instead of that blastocyst stage embryo we're talking about, which in humans has about 100 cells, if you use an even earlier embryo, like this is an actual picture of a human embryo that has about 8 cells, you can actually pluck off one of these cells and use just one cell, and that cell is sometimes called a blastomere. And just from one cell, sometimes if you do it just right, you can grow an embryonic stem cell line from that one cell. And remarkably, the other 7 cells, the rest of that embryo that has, say, 7 cells or sometimes maybe only 9 cells left, is still viable. It actually still can function and develop. And you can freeze that embryo back down if you want. So this was reported in Nature back in 2016, that you could do this so-called single blastomere method. So ESCell research, and we sometimes call embryonic stem cells ESCells for short, has sort of a long, very distinguished history. And it actually started, for the most part, using mice as a model system. And a number of researchers who discovered that you could make embryonic stem cells from mouse embryos and use them in different ways to make, for instance, mouse models of genetic diseases and things, actually won Nobel Prizes. And this included Mario Capecchi, Martin Evans, and Oliver Smithies. And I just go through in this post that another researcher, her name is Gail Martins, I think actually deserved a Nobel Prize too, because she did some of the pioneering work in this area as well. But unfortunately, the Nobel Prizes have sort of this history of being biased against women. So after the embryonic stem cell work was done in mouse in the early days, it was back in 1998 that Jamie Thompson was the first to successfully do this with human embryos and grow out human embryonic stem cells. And that's sort of the history there with human embryonic stem cells, which are now, more than 20 years old as a research field. So because human embryonic stem cells are most often made from embryos, there's been sort of debate about, is that ethical research? Should that be legal? And so I often get asked these days, is embryonic stem cell research legal? And the answer is, today in the US, it is actually legal at a federal level. NIH can also fund that research. So that's kind of the big picture answer there. It's also notable, however, that within the US, there are certain individual states that have laws about embryonic stem cell research that kind of restrict the kind of work that can be done within the borders of their states. It's also something that's sort of striking to mention is that the NIH currently lists almost 500 human embryonic stem cell lines on its website. You can go to my site here to get to that link with the NIH list of embryonic stem cells that they have in their registry. Now, of course, there are a variety of different laws around the world in different countries. Some countries make embryonic stem cell research not something that the federal government in that particular case will fund. Other countries are restrictive about embryonic stem cell research regardless of the funding source. So now let's shift gears a little bit and talk about how you can actually use embryonic stem cells in research. So there's a wide variety of things that you can do with embryonic stem cells. You can differentiate them into different kinds of cells. And I wanted to show this video that's really cool of human beating heart cells that were made in my lab from human ES cells. And hopefully you'll be able to see this pretty well. And it's actually pacing along and beating almost at about the same speed as a human heart would beat. So this is essentially like a little piece of a human heart made from embryonic stem cells in a dish in the lab. So you can imagine if you can make these beating heart cells, you can do a lot of heart research, right? And if you can make brain cells from these ES cells, you can do a lot of brain research. So there's a ton of things that we can learn about human development and human diseases from using human embryonic stem cells. Of course, in more recent years, another kind of cell called human-induced pluripotent stem cells, or iPS cells, have come onto the scene. And these cells are basically equivalent to human embryonic stem cells in basically almost every way, but don't need an embryo to be made. And so they can actually be made from a patient's own skin cells or other kinds of cells. And the second potential bonus about iPS cells is that if you make them from the potential patient themselves, then they're a perfect match to that patient, right? You don't have to worry about if you were to use the iPS cells to, say, make eye cells and then put those eye cells into a patient's eye. If the iPS cells came from the same patient, their immune system won't attack those cells. Both embryonic stem cells and iPS cells have the potential to form this kind of tumor called a teratoma, which is kind of an interesting tumor that can have all different kinds of developmental features and different kinds of cells and tissues. Here you can see these almost like sea anemone-like structures are actually human glands. They might be mucus glands. There's different connective tissue in here. There could be even some muscle, potentially some brain. And so these teratomas are actually really interesting from a research perspective, but you wouldn't want them to develop in a patient who gets a human embryonic stem cell or iPS cell-based treatment. So it's really important to get rid of any remaining ES cells that are not differentiated before you transplant anything cell therapy-wise into a patient. And if we look at the different clinical trials that are going on with embryonic stem cells, and I currently found 45 on clinicaltrials.gov, the database that mention human embryonic stem cells. Again, you wouldn't want to use the embryonic stem cells themselves. You want to differentiate them, say, into pancreatic cells, like work that's being done by ViaCyte or by other companies, and get rid of any remaining pluripotent cells before you transplant them in. And for instance, in the work by another company called Lineage Cell Therapeutics, they're trying to develop, and they're testing actually a product that could potentially help spinal cord injury. So the overall conclusion here is that these cells are really important both for research and medicine. They're legal to use. I think they're ethical to use. The recent polling I saw in the US actually showed that about 2/3 of Americans view human embryonic stem cell research as something that is ethically okay to do, which is a change from past years. There's still some debate, in different quarters about that, but I think it's reassuring to see that the vast majority of Americans now are supportive of that work. I've got some references here in this blog post if you're interested in checking it out at ipscell.com. So I think we've covered all the main parts of what I wanted to tell you about human embryonic stem cells. Let me know if you have any more questions. Again, this is Paul Knopfler, professor at UC Davis School of Medicine. And I'll see you next time with another video about different things related to stem cells. And I'll see you later. Bye-bye.